I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley. I study how nanoparticles can be functionalized synthetically to be useful for biological applications, ranging from imaging to gene editing. What drove me into science was curiosity, and the bulk of my training has been in curiosity-based science. I was curious as a child. Growing up, I really liked testing things and probing things, and I wanted to be extra sure and extra careful that something that I came across was validated, or that the data held up to my hypotheses. My upbringing in living in many different places and traveling to many different places has helped me contextualize the world as something that remains to be explored. A part of what I really enjoy about doing science is that it also is something that remains to be explored. Having my parents be in the education system, having them both be teachers now, and having my mom be an engineer as a role model motivated me to consider that as a career option as opposed to just a hobby. When I started my undergraduate career, I was pre-med for maybe an hour or so and realized fairly quickly that I was interested in more engineering approaches to biological sciences as opposed to the biological sciences themselves. I think I was fortunate to train in fairly basic fundamental sciences, physics and chemistry, which allowed me to have an appreciation for the laws by which matter in the universe abide. What I'm passionate about now is exploiting that training to engineer or make tools that can then turn around and probe those fundamental interactions. The focus of my research is to develop nanoscale probes that can be used either to image molecules in biological systems or to deliver functional biomolecules to biological systems. The importance of this work is in our ability to either see things that we wouldn't normally be able to see in biological systems, or to deliver a functional biocargo to systems such that we can engineer those systems genetically to be more desirable. One question that I'm trying to answer is how antidepressants and antipsychotics are affecting the kinetics of neurotransmitter release and reuptake in the brain. So we have developed probes that are selective for a neuromodulator known as dopamine, which is a common target for antidepressants and antipsychotics. On the one hand, we're studying how the nanomaterials that we use might be influencing the role of microglial cells, which are cells that are the resident immune cells of the brain. And then for brain chemistry, we're really interested in seeing what the effects of currently available dopamine reuptake blockers, such as antidepressants, are having on brain chemistry, how those are affecting the kinetics, and really just zooming into different cells in the brain to see why there might be so many negative side effects of using antidepressants or antipsychotics, why these drugs tend to have very high patient-to-patient variabilities, what we might be able to do, again, from a molecular perspective on increasing the efficacy of the drugs and decreasing their off-target effects. The other big question that I'm trying to address is how we can functionalize nanomaterials to better deliver functional biological cargo for gene editing in plants. When we talk about genetically engineering plants, the end goal is not necessarily to give them superpowers, but to make them able to live in a changing global climate and reduce land mass on which to grow crops. So it's not so much a matter of them becoming super plants so much as it is kind of resuscitating the diminishing quality and quantity of plant and agricultural matter that we're producing. Even broader than that, my goal is to produce science that is interesting, insightful, and useful. And the reason that I have those goals specifically as a professor is that I want to be in the pursuit of those goals with students and postdocs. I'd say both of those aspects of this job contribute to how much I really enjoy it. So I want to produce good science, and I want to produce good scientists in the process too.